The next presentation is from Anna Terrade from France, and we've got that on recording and online.
Hello everybody, my name is Anna Terrade. I'm a PhD student, and the objective of my research is to find an acoustic signal that will keep animals away from tracks in order to reduce wildlife train collisions. This project is a collaboration between the French railway company, the SNCF, the ENES Bioacoustic Research Laboratory, and the French National Museum of Natural History. And today, I will answer to this question: do ultrasound elicit a flight response in red deer hinds? Each year on the metropolitan French rail network, wildlife train collisions cause important financial and ecological costs. And significant incidents occur mostly with large ungulates such as wild boars, red and roe deer. In another part, ultrasound devices are largely commercialized to avoid deer vehicle collisions, for example, despite their poor propagation properties. And because they are inaudible for humans, we think they could deter animals without disturbing us. But in reality, we know that deer and pigs show an hearing range close to ours, which will only be reliable below 30 kHz. And even if previous studies show poor efficiency of ultrasonic frequency on ungulates, they were mainly focused on pure tones, and here we wanted to test ultrasonic frequency modulations. So here, our focus species is red deer hinds, and because Pecarries showed a surprisingly strong flight response to our pilot study, we chose them as the contrast species. Each species, respectively 29 individuals divided into 5 groups for red deer hinds and 10 individuals for Pecarries, were exposed to sequences of 9 ultrasonic frequency modulations between 20 and 30 kHz, ended by an audible signal as the control sound. And here I have lowered the frequency of a sweep stimulus as an example of these modulations. Okay, so each group were exposed to multiple playbacks, and I have observed their reaction in the field, but I've also recorded them with cameras in order to analyze their behaviors at the lab. And I have observed detection, vigilance, and avoidance. Okay, so here are my results for red deer hinds, and I did not observe a flight responses in reaction to playbacks of ultrasonic signal over the 20 playback trials, as you can see in this video. So here the hinds are just eating, and the only reaction we could observe is due to dominance, here, as you can see. And for the all male trials, the only behavioral reaction were detection and vigilance. And we can see here in this graph that statistically, responses of red deer hinds are significantly higher for the audible sound than for the ultrasonic stimuli here. So for Pecarries, I have observed a strong flight response at each onset of my sequences, indicating they can hear ultrasound even if no studies have shown it yet. As you can see in this video, we can see the flight response here of all the group. And during my in-situ observation, I saw a short-term avoidance of the speakers after stimuli, but also that individuals get habituated to stimuli within the sequence. And when we compare the responses of Pecarries compared to red deer hinds, we can see that they respond more to ultrasonic stimuli than deer, except for the audible sound. And this difference can be explained by the habituation of Pecarries because the control sound is at the end of the sequence. To conclude, ultrasound appeared to be inefficient for inducing a flight response in red deer hinds. And this is not surprising, since are likely to have evolved aversion to ultrasound because these mammals vocalize in the audible range, which explains their reactions during the audible control signal. Moreover, the poor propagation properties of ultrasound make them technically difficult to apply as deterrent signals. And also, they are very poor candidates for reducing wildlife train collisions. That's why I will continue my work on audible sounds during the rest of my PhD. So thank you to all my colleagues who worked with me in this project, and thank you all for your attention.
Thank you, Anna. Now, I understand you're there standing online for some questions. Any questions for Anna? Let me just say as a statement, it's always good to get negative information, to present negative information. We need to learn from the things that don't work as well as the things that do. Any questions for Anna? Right.
Anna, can you hear me? Yes, you can hear me?
Yes.
Yes. I don't know I know nothing about these techniques, but according to you show that the animals were able to accustomate with the signal. Should a modification of the signal each time avoid this accustomation of animals to the signal and would have more efficient results?
Yeah, here for the ultrasound experiment, it was always a new stimuli within the sequence. But for my research and for audible sounds, I will try to have different sound with a random resynthesize. They will be created each time randomly to avoid this habituation phenomena.
Thank you, Anna. There's a question from the floor.
Thank you, Anna. I think you answered my question there, but it was going to be, what are the next steps? Because there's lots of, I think we would like noises to work, but often they don't. So what's next, and what do you think the possibilities of an effective solution is?
Yeah, I know it's a complex subject because a lot of things have already been studied as a gas exploder or other things for files, for example. And here my idea is to find maybe acoustic characteristic in alarm and distress calls of animals. It could be a perfect. It will be meaningful for my species. For example, if I take an alarm call from a deer, it normally would enhance a response for the conspecific species. Sorry. But here the main problem is the heterospecificity of my species, because as I've said in the presentation, I have wild boars, roe deer and red deer. And the idea is to find the acoustic characteristic in their distress and alarm calls to maybe resynthesize them and use them to scare them from the train. I don't know if it's...
Yeah, thank you very much, Anna.
Yeah.
Thank you. All right, we'll move on to the next speaker. Thank you.
Okay, thank you.